Thank you. Yes, good morning. Thank you to the conference organisers for this brilliant conference, which I've found really useful and really interesting, being relatively new to the microglia world. And thank you for giving me the chance to talk about our work. So I'm going to just take a few slides initially to explain the process we use at Alchemab to discover antibodies, and then focus in more specifically on an antibody we've discovered against CD33, which we're developing. So we don't start with the usual target-first approach to drug discovery. We instead start with patients or individuals that we classify as resilient. And that resilience can look different depending on the disease that we're interested in. But you can imagine, for instance, something like oncology. It might be somebody who's been diagnosed with, say, pancreatic cancer, which usually has a median survival of less than a year. But there are some very rare, exceptional individuals who survive 7 years or more, 10 years or more. And what is it about them that gives them this survival benefit? It's a little harder to find resilience in neurodegeneration, but it might be, for instance, somebody with a genetic susceptibility, and we've heard lots over the course of the few days about that, or somebody who might have biomarkers such as amyloid, but don't progress on to have tau or neurodegeneration, or who don't progress beyond mild cognitive impairment. And then we have the ultimate resilient people, the supercentenarians who've lived over 100 years in good health, no major chronic health issues, and still with excellent cognitive function. So what is it that keeps these people well, and these and other resilient people? And at Alchemab, we think that at least part of the answer to that might be antibodies. And we're used to thinking about antibodies in the context of infectious diseases, where are B cells, and we have the B cell receptors. When they become plasma cells, they produce antibodies, which are the soluble form of the B cell receptor that binds to pathogens, and they're cleared away as part of our immune response. Because any one individual may come across so many different pathogens in their life, we produce an awful lot of different antibody variants, or we will have the capacity to produce a lot of different antibody sequences. An individual can produce up to 1 × 10⁹ different antibody variants. That means that we do have this great width of protection against different pathogens we might encounter. But it also means that sometimes, just by chance, we also produce antibodies against self-proteins. And we're more used to thinking about them in the context of autoimmune diseases, where those self-proteins and the binding to them may cause pathology. But what's to say there's not a flip side to that, where we can also have self-antibodies that are protective? And this does happen. Here's an example in breast cancer, where breast cancer patients naturally produce anti-HER2 antibodies. So we develop anti-HER2 antibodies as therapies, but some people naturally just produce them. And it does confer a survival benefit. You can see the red line there is these patients that do have these HER2 antibodies, whereas the blue line is patients that don't. So they do exist, these protective self-antibodies, and we think that they probably exist far more than we realize, and in other diseases, not just oncology, but lots of different indications. So what we do is, it all starts with patients. We have to get hold of fantastic cohorts of samples, and we group them together as resilient, but also non-resilient, so progressing populations, and also healthy controls. We then carry out deep sequencing on their B cell repertoire, so on the B cell receptors, and this gives us antibody signatures for these individuals. And then we look for antibodies that are shared amongst these resilient people that aren't found in the progressing populations. And it's worth noting that's very rare to have the same antibody as somebody. The odds of you having the same antibody as the person you're sitting next to is very low, unless you've been vaccinated against something. So we probably do all share kind of quite common antibodies to something like COVID-19, but beyond that kind of selection pressure, it's very unlikely that you'd have the same antibody. So to find that same antibody in 2 or more resilient people and not in the progressing people is actually a very strong and very compelling signal. So we're at a microglia conference, obviously, and this is because we found an antibody through this process which binds to CD33. Something we've heard a lot about in previous talks, and we first identified this in a cohort of patients from the European Prevention of Alzheimer's Dementia, or EPAD, which was a brilliant cohort of patients and samples which came with a wealth of biomarker data as well. So we were able to classify resilience based on this kind of ATN profile first described by Jack et al., which talks about Alzheimer's as a continuum, which starts with amyloid, leads on to tau, and then ultimately to neuronal injury. And we were interested in these individuals in the pink box who had amyloid burden, but didn't progress on to having tau or to having any cognitive impairment or neuronal damage. So using this classification of our resilience, we ran these samples for our usual sequencing and bioinformatics pipeline, and out of the end came 96 antibodies of interest, which were shared between 2 or more of these resilient individuals. And one of them we found bound to CD33. So in the top left there, top right from your direction, you can see that the ELISA data, that one little lonely dot there, that was the one out of 96 antibodies. It wasn't a terribly strong binder, but we were excited enough in this as a target, knowing its links to Alzheimer's disease, that it inspired us to look in other patient samples as well. So we have multiple cohorts, different neurodegeneration diseases, and also our supercentenarians. And we actually found CD33 antibodies in quite a few of these resilient individuals. That allowed us to then conduct a phage display campaign, out of which came a whole panel of patient-derived anti-CD33 antibodies, which we screened through various kind of cell-free and cell models, and ultimately settled on our lead antibody of ATLX1088. So we've all heard about CD33 already. It was a fantastic talk yesterday, which did a much better job than I could in explaining its relevance. But just to recap and to explain why we were so interested in this when it came up as a target. So CD33 is an inhibitory receptor. It's expressed on microglia in the brain, but also monocytes in peripheral blood. And it's an inhibitory receptor. So it binds to sialic acid, amongst other ligands, as we've heard. And its expression is increased in microglia in Alzheimer's disease patients. And the more of it there is, the more advanced cognitive decline. It's often talked about in context of its relationship with TREM2, something else we've heard a lot about. And you can kind of think about them as the activatory receptor being TREM2 and then the inhibitory receptor being CD33. And there are lots of GWAS studies linking it to late onset Alzheimer's disease. There's a protective allele, which due to differential splicing loses the exon 2, which at the protein level means that it loses the ligand binding domain. So the part that binds sialic acid is no longer present, so it's effectively non-functional at this point. It also leads to less of the kind of full-length protein being expressed on the cell surface. So all of this together tells us that reduced CD33 expression and also reduced activity is protective in Alzheimer's disease. So how we imagined that an antibody might be having a protective effect is if we think about amyloid plaques, which we know are coated in sialic acid. So if an if a microglia approaches a plaque, it encounters sialic acid, which binds to CD33 and has an inhibitory effect. If an antibody instead, like on this other image here, was to block that interaction of the sialic acid and the receptor, that would then remove this suppression. Phagocytic function would be restored. Microglia could then go about their job of clearing away the amyloid plaques. So that was our hypothesis. We wanted to test it in a relevant cell model. So we used iPSC-derived microglia, and we have a live cell imaging system called an IncuSite, which you can take images of every half hour, every hour, however often you like. And we used amyloid beta as bait. So we labeled it with Frodo Red, a pH-sensitive dye that only fluoresces once it's taken up inside a cell. So we were able to measure red signal against time as a measure of phagocytosis. That's what you can see on this plot here. There's a trace of time against red signal. And the green triangles down the bottom are microglia that were treated with isotype control antibody before being exposed to the bait. And then the red circles are microglia that were treated with ATLX1088 before being exposed to the bait. So there's a considerable increase in uptake of amyloid beta in those cells. Some of the other traces down the bottom are other anti-CD33 antibodies. So 5810 is another Alcomab one that came through the pipeline. We know it binds CD33 well, but it doesn't have this same kind of impact, so it's not enough just to bind. And likewise, AL003 is another anti-CD33 antibody that got into phase 1 trials, but in this assay, at least, it didn't have the same impact as our antibody. So these plots are from that same kind of experiment, but just displaying it now as an area under the curve rather than the total trace. And in green, you can see microglia treated with the isotype control antibody. And then the two different blue shades of blue on that first plot are two different batches of the antibody. So this was very encouraging to us to see that we can make this stuff more than once and see the same impact. And then we also were interested in seeing the impact of TREM2 antibodies in this same assay system. And it was quite varied. The three antibodies we tested, one seemed to maybe slightly decrease phagocytosis, one didn't have too much impact, and one did increase in a similar way to ATLX 1088. And then the other kind of part of CD33 biology we were really interested to explore was how it internalizes when antibody binds. Because this is a common thing you find with anti-CD33 antibodies. They quite often cause rapid and significant internalization of the receptor upon binding. And this is something that's actually been exploited in the oncology field. In acute myeloid leukemia, they express CD33. So it's a fantastic target for getting antibody drug conjugates specifically into a cell. Maybe not so great in neurodegeneration when we want to get the antibodies to the brain, because it's also expressed on monocytes in the blood. So if we inject our antibody, it binds to monocytes, it internalizes into them, it's being taken out of the system before it gets a chance to get to the brain and act on microglia. So we're very interested to see what our antibody did for internalization. And this plot is some flow cytometry data. We're looking at CD33 at the surface of the cell. So internalization, we kind of see a loss from the surface. We'd infer that to be internalization. So decreasing values on this plot represent internalization. Along the top in blue, you see the isotype control antibody, so you're not seeing any loss. And then down the bottom in purple and a greeny color, they're kind of overlaying each other. There's gemtuzumab, which is one of those oncology drugs which we know does internalize very rapidly, and also AL003, which was also designed to cause very rapid internalization. So we do see that loss there. But we were surprised and pleased to see that actually our antibody is tracing along up there with the isotype control, so in the pink there. So at least at 5 hours, so at an early time point, we don't see any loss. It does start to internalize later on, but in these early hours when we're worried about it being lost in the blood before it can get to the brain, it doesn't seem to be causing internalization, which is quite unusual for a CD33 antibody. And we believe it will give us a pharmacokinetic benefit in that we'll be able to get more to the brain. So testing this in vivo has some interesting challenges. CD33 biology is not the same between human and mouse, and even if it was, our antibody doesn't cross-react with mouse. So we found it in patients, we found it in humans, and it's not cross-reactive, and we didn't want to engineer in any cross-reactivity because we want to keep it as close as possible to what we actually found in the individuals. So we needed to carry out PK studies in humanized CD34 mice, and it was exactly as we would expect for a standard IgG molecule. So there was no suggestion of a peripheral sink or of this loss of antibody into the monocytes, which is very encouraging and tied in nicely with what we'd seen in the in vitro data, showing that we don't have this internalization. For efficacy, we need a more elegant model. So we're very lucky to be able to access the model at the VIB Center for Molecular Neurology, where they have iPSC-derived human iPSC microglia implanted into the APP NLG, is it, model of Alzheimer's. So we were able to see the impact of our antibody on human microglia in an amyloid context. So that is ongoing at the moment. We're very interested to see what might come out of that. So finally, just to kind of leave with this point, I've heard a few times over the course of this conference the phrase, our favorite receptor, TREM2. So I'd just like to put forward for your consideration the idea that maybe CD33 could also come up there as one of your favorites too. There's genetic support for both of them. It may be different approaches. Possibly it might be that you'd want to look at agonism for TREM2 versus antagonism for CD33. You can imagine that might result in the same kind of outcome. In terms of the type of antibody, we've gone for a standard IgG, so not engineering too much into it. The only thing we have changed is that we've removed the Fc effector function because we believe this will confer some safety benefits. And then we're not into the clinic with this yet, so we have no information around efficacy, safety, biomarkers, that kind of thing. But we're hoping we can learn from the TREM2 antibodies that have gone into the clinic in terms of biomarkers that we might be able to utilize as well, and any kind of safety things that we may need to look at, as Aria just mentioned it in the previous talk. So yes, that is my final slide. Other than to just thank the whole Alchemab team. Obviously, it wasn't just me who worked on this. And that is pretty much the whole Alchemab team, almost, outside one of our sites in Whittlesford, which is just down the road. And there's also some of us on the Bagram Research Campus and a few in Boston, in the US as well. Our collaborators who provide the samples, without which we can't do anything, so very, very critical people. And then our funders there at the bottom. Thank you.
Great, thank you, Helen. Great talk. I think we've got time, certainly, for a few questions. So I think down here. Where are the microphones? Yeah. Do you want to start over here?
Hi, really great talk. Just something moving a little bit aside from the neuro part. When you were looking at the monocyte binding of the anti-CD33 antibody, did you also look at the whole PBMC fraction, so consider things like NK cells, and if you saw any activation of the NK or any degree of phagocytosis due to the FC portion of your antibody in response to treatment?
Yes, so we did look at total PBMC fraction a while ago. We didn't, in terms of kind of binding, there was some binding to other cell types because it is expressed on some others. We didn't particularly look at phagocytosis itself. We only looked at it in monocytes, but we looked at things like cytokine release to see if there was any concerns from more general activation of PBMC. And it's incredibly clean in terms of cytokines to the point at which I was thinking, does this antibody do anything? Because I kept putting it on cells and then nothing was coming out. So yeah, not specifically phagocytosis, but we have looked for more broader effects in other immune cells.
Thank you. Well,
do you see activation of the item motifs with your antibody, and how does that compare with the other anti-CD33s?
So that is something we're working on at the moment. Our protein biochemists, I know, are wrangling with it. It's a little challenging, the kinetics. But yeah, that's definitely something we need to find out.
great, one more up here.
Have you sort of excluded binding to A-beta, and at least whether in part the mechanism might be sort of opsonization of A-beta? Or does it solely bind CD33?
We haven't excluded it, actually. No, that's a very good point. No, and that should be fairly straightforward to do. So yes, I will do that. Thank you.
Good. Yeah, okay, one more, Angela.
Really fascinating work. I just wondered if you could tell us a little bit about what you think the initial immune response in these antibodies produced by people. Would they have had amyloid angiopathy, maybe? Or you said you had other hits as well, which I don't know if that tells you anything about how the immune system has created these antibodies in the first place.
Yeah, it's a really kind of critical question. And we have some metadata on patients, so we look for other sort of comorbidities or other things they might have which would be driving it. There was no obvious signal in that sense, and the question does arise as what it rose against originally. It was probably peripheral CD33 to be most likely, and then just by chance that has a benefit in the brain. So that's how we're thinking at the moment.
I guess that was kind of related to my question then. So what about other targets that we might be interested in then if they don't have a peripheral footprint?
Other targets that might be peripheral?
Other antibodies that we might be able to find would potentially need a peripheral target then?
Yes, that's always a possibility with the sort of the neurodegeneration cohorts, thinking, will we find antibodies against something that's just in the brain only? For some other projects, we have found antibodies against more brain-specific targets, so it's not that it can't happen. But in this instance, it seems, at least 50-50 chance that it was peripheral, but we can't say for certain.
Okay, maybe one last question for me. Can you share any thoughts on why you think it doesn't lead to the internalization in the monocytes, or is that still TBD?
Yes, it is something we're working on, and it was surprising. And there are questions around whether maybe it does, but then just very rapidly comes back to the surface because CD33 kind of does recycle quite quickly. So we're doing some imaging studies looking at internalisation, then where it goes afterwards. But yeah, we haven't figured that out yet.
Fair enough. Thank you. Thanks very much, Helen.
Thank you.